Welcome to our October virtual coffee hour. My name is Agnieszka Carpenter, and I'm the Executive Director for BioMe. BioMe is Bioscience Association of Maine. We're a trade industry group devoted to growing and advancing bioscience in the state of Maine and beyond. And the BioMe virtual coffee hour is a now bi-monthly series where we hear from 4 experts, 4 amazing speakers, talking about research, new developments, all sorts of associations, services, and other developments in the bioscience community in Maine. And before I introduce the speakers, I also wanted to thank our sponsors, and of course it's not working. There we go. These are our annual sponsors, and without these amazing organizations, we wouldn't be able to put together events like this one or a lot of our in-person events that we're very happy to be organizing at this point. If anybody's interested in sponsoring us in 2023, now would be the time to contact us. So just a shameless plug in here, but we love our sponsors and appreciate everything they do. And a couple of logistics before we jump into the meeting. The chat function is on, so feel free to use the chat either to ask questions or to just say hi. We do have 10 to 15 minutes of Q&A planned towards the end of the meeting. And if anybody would like to ask any questions in person, just raise your virtual hand and I can let you do that. And each presentation will be 10 minutes long. And we're not stopping in between. We're saving the questions till the end. If you've done coffee hours before, I apologize for being repetitive, but I do see some new faces. So I'm sure not all of you are familiar with this format. Okay, so not to waste any more time, I would like to introduce our first speaker today. And our first speaker is Dr. Dorothy Klimis-Zacas from Maine, who's the professor of clinical nutrition. And Dorothy, whenever you're ready, share your screen, please. And I need you to unmute.
Unmute. Good morning. Hi, and thank you all for inviting me to speak today. And I'll just take 10 minutes and go over a kind of review of some of the projects I have been working on. I have worked with wild blueberries for a long time. If this can move. Okay. Should I put it on presentation mode? Probably like this. Okay. And wild blueberries are very, very interesting. They have hundreds of bioactive compounds that have health promoting benefits. And I have been working both with wild blueberry enriched diets and with extracts from wild blueberries. And the areas of research I have targeted usually chronic disease, cardiovascular disease, metabolic syndrome, and more recently, wound healing. And we have targeted vascular function and structure, metabolism, lipid and glucose metabolism, inflammation, and more recently, cell migration and angiogenesis. So I will just very briefly go over some of this, the findings. I have worked in preclinical and clinical studies with different types of animal models, normotensive animals, hypertensive animals that represent the metabolic syndrome. Also clinical studies with subjects with multiple risk factors for cardiovascular disease, and also worked in vitro studies with cuvets. So there is this concept that I have coined, endothelially targeted nutrition. And because with diets, we see, and especially with the wild blueberry diets, we see effects, immediate effects on vascular function and structure. And pharmacologists have been trying to do this to target the endothelium with vascular directed agents. So we're using this unique apparatus that pharmacologists use, the retinotic tissue baths, okay? And what we do is to study vascular function. We place animals in different diets. We harvest after a while that they're being fed, usually 6 to 8 weeks, harvest their aorta and we slice them in rings and place them in this rhabdotic tissue bath and in an ex vivo situation. And force transducers will be able to then detect and measure maximal force of vessel contraction and relaxation after we challenge them with phenylephrine and or acetylcholine in a dose response curve and calculate vessel reactivity. And so these are the force transducers that we have been using, unique in the area of nutritional biochemistry. What did we find? Well, in normotensive animals, we find that wild blueberries prevent or reduce vasoconstriction. In spontaneously hypertensive animals, we find that wild blueberries lower vasoconstriction, but also potentiate greater vasorelaxation. And in obese Zucker rats that are models of the metabolic syndrome, and they have abnormal vascular dysfunction, we see that blueberries normalize that function. Very important findings, all published. Then we ask the question, well, how do the blueberries operate? Well, we discovered that they preserve nitric oxide bioavailability, or one of the vasodilators, and they interact and work on all the steps of the nitric oxide, guanylate cyclase, and cyclic GMP to improve vascular tone. Okay, I don't have time to, but it's the smooth muscle cell they operate through with preservation of nitric oxide bioavailability and protection. So a wild blueberry diet, and just 8 weeks, about a cup and a half or a cup, can be a preventive and therapeutic strategy. It's not expensive. Okay. And then we went on and we said, well, vascular function change, what happened to the structure? Did the structure change? So we targeted this molecules, complex molecules, glycosaminoglycans. Okay. And again, pharmacologists are targeting this with special attention to chain elongation and sulfation patterns. So what are those? Well, usually they're part of the connective tissue, but they form the glycocalyx on the intima of the artery. And they are, you can see the molecules, the syndicans, the glypicans. They're usually embedded in cell membranes and lipid rafts, okay, as you can see here. And they are mechanotransducers. They're just like biosensors, amazing molecules. And so what we found that after we fed the animals for 6 weeks, that these were the spontaneously hypertensive rats, abnormal distribution of these GAGs. So we discovered after we fed them that the GAG populations, the antitherogenic increase and protherogenic decrease. And also, most importantly, we discovered alteration in sulfation pattern. So we found decreased sulfated and increased non-sulfated GAG. So this implies that the LDLs are less likely to dock on the endothelium. So this was something very interesting. So the blueberries to be able to reduce the ability of the vascular cell to retain lipoproteins and initiate the process of atherosclerosis. After that, we went on to study obesity-induced inflammation and the metabolic syndrome. Again, you probably do know what metabolic syndrome is and the different characteristics of insulin resistance, hypertension, obesity, and dyslipidemia. So we use this animal model, the obese Zucker rat that mimics and its similar characteristics and progression to the human, where they are hypertriglyceridemic, hyperlipidemic, cholesterolemic, insulin resistance, and they're inflamed. So we placed them on the blueberry diets and studied inflammatory markers, gene expression, lipid profiles, and blood glucose regulation. And after 8 weeks, the plasma, again, the obese Zucker rat has everything abnormal. So plasma circulating markers of inflammation decreased, and expression, of course, adiponectin increased, and expression of inflammatory molecules was significantly downregulated both in the liver and the adipose tissue. Again, another thing that we observe, which is consistent, all these things have been repeated and they're consistent observation over 15 or 20 years. The wild blueberry consumption decreased dramatically triglycerides and non-HDL cholesterol, and also key enzymes and transcription factors that are involved in lipid metabolism, so that the blueberries help increase oxidation and decrease accumulation of fat. So all those taken together tell us that blueberries have an anti-inflammatory effect and a lipid normalizing effect. So it's a safe, effective, and economic way to perhaps prevent or reverse. Then they have done clinical studies in collaboration with the University of Milan. Going there as a Fulbright fellow and a Cariplo fellow. For many years, they have this collaboration for the last 15 years. And we study vascular function. I do my clinical studies there, study vascular function and so many other parameters. I'm just going to touch on it. On 6 weeks, this is a crossover design that we place the group of patients. So the total thing studies like 18 weeks, and we measure different parameters. Very, very comprehensive study. And we use the EndoPath, which studies vascular function in humans. And we find that vascular function improves in subjects with endothelial dysfunction. There is a protection of DNA against oxidation in acute studies. You see as the anthocyanins increase in the phenolics in the bloodstream, and so does DNA protection. So, and then finally, most recently, I am involved with in vitro studies and exposing and utilizing wild blueberry extracts to see whether they're going to promote or inhibit migration and angiogenesis in HUVECs associated with wound healing and tissue regeneration. Angiogenesis, as you all know, is formation of new capillary blood vessels from pre-existing ones, and the goal of the study was to investigate different fractions from the blueberries as to their role. So we extract from wild blueberry powder phenolic acids and anthocyanin in different fractions and expose HUVECs and study to them and study the speed of endothelial cell migration and angiogenesis, whether they are able to form endothelial network. And also gene expression critical for migration process, RAC and RAW, and genes critical for formation of endothelial network, VEGF, ENOS, ACT. And so what we find, I have videos on this, but we don't have time. So what we find when we expose these cells, we see that the particular concentration, this is the control, the wound or the scratch in the ACN in anthocyanins does not close even 8 hours, but phenolics are already closed at 6 hours. Combination also does. So we can see ACNs inhibit cell migration and promote, phenolics promote it. And when we do look at the network of blood vessels capable to be formed in the control, you see the nice network. ACNs, they just fall apart. They cannot. The phenolics at a particular concentration, the form network, and so does the combination. So that was all interesting because we discovered this differential effect of anthocyanins and phenolics on migration and angiogenesis. Also the gene expression of all these different proteins reflected what we found. It was upregulated with the phenolic fraction. So we decided, let's use the phenolic fraction and go one step farther, because this can have implications for wound healing, diabetic foot ulcers, burns, ulcus decubitus, and any condition that can benefit. So we went on, and so we published this, and we also applied for patent. And we went on and did preclinical studies with animals with the blueberry extracts first, whereby we just used the phenolics because we wanted to check for promotion of this migration and angiogenesis. And as you can see, we created 4 dorsal wounds and applied different concentrations, the phenolic acids and the mix of phenolics and anthocyanins. And you can see here, we did this during COVID, very, very difficult. Created the dorsal wounds in the animals and just adding the extracts. You can see them we photographed every day and measured, and can see control. It was 6 days, just once we applied it, once per day, and we see the wound closure and was about 20%. and P500 and in the mix. They all did well. But looking at the other parameters, our inflammatory markers, our angiogenesis markers, we chose to use the best performance was the phenolics 500. So we just continued with phenolics, and with an industrial partner, we formed a prototype. And the prototype was either a serum or a cream, okay? And we conducted similar experiments with the preclinical studies with just the two concentrations, applying them. And we discovered that serum 500, it was about 12% wound closure above and beyond the control. The cream was not significant. And then we started many parameters. We don't have time now to show you. Vascularization, we saw again with the serum 500, we had very high vascularization, collagen formation, immune response, all is very positive. And so we had see 12% increase in wound closure, 20% increase in cellular vascularization. But this is a very hopeful product that we can bring to market eventually to help these individuals with medical conditions such as diabetic foot ulcers, venous ulcers, burns, post-surgical wounds, etc. And it's low cost, no side effects, easy application, just once a day, all natural nature to improve quality of life, reduce hospital stay, and decrease medical expenses. This was my team, my graduate students, Panos and I were co-inventors. He's now at Johns Hopkins, and Natalie, who already has her doctoral and working for a biotech company. And we were involved in the entrepreneurship, customer market discovery through the University of Maine I-Corps and the National I-Corps, the MARTA, the I-Trap, the funding came from different sources. And of course, I have the Maine Institute of Technology level 1 and 2. And of course, I thank all my students, hundreds of students, and that have helped me along the way. So that's it. That's all. I did it. It's done a minute. All right.
Thank you so much, Dorothy. I think what I got from your presentation is that we should all just keep eating wild blueberries and put them all over our bodies. But we probably, a lot of us have questions about how we can apply this to our daily lives. So we'll leave those questions till the end. And now if you could stop and share. We will jump to our next speaker. Our next speaker is Marty Grohman, the Executive Director of E2 Tech. And I've been wanting to get Marty on for quite some time, even though E2 Tech is not necessarily bioscience, but I think it's great to know what other associations are doing, and energy is such a big topic right now. So Marty, whenever you're ready. And we can't hear you yet.
Hi, I'm Marty Grohman, Executive Director of E2 Tech, Maine's largest community of energy and environmental engineers. Thanks for having me. I'm a little less techie than Dorothy, and not as interesting, but I'll try. So we've been around about 20 years. We've been growing. I took over as Executive Director almost 4 years ago now, and we're about 200 companies, so I'm proud to show that number to you there. We have about 5,000 individual members across all those companies. That number is basically fixed because the next tier of our membership software, that's when the price goes up at 5,001. So we do weed out emails when we get bounce backs. But we are a convener connector. I'm forever just pulling together people from government, from industry, from startups to help promote them. And E2 Tech does not lobby or advocate, but we do push our sector, which is clean tech, climate tech, renewable energy, environmental engineering. So I don't ever go up, even though I'm a former legislator, to Augusta to say, you know, pass this solar bill or don't pass this PFAS bill, but I will get involved in promoting our companies and helping them grow. And we do pride ourselves on doing a lot of events, which we always make free for state of Maine employees, students, and elected officials. So we often get a good convening of people and offers. It's kind of a roundtable discussion that's helpful. And we had a lot of ties to the startup world too, which I will want to talk about. These are the companies that make what we do possible. Dorothy mentioned MTI. We're pleased to be closely affiliated with MTI, and I suspect everyone here knows how to reach MTI, but if I can help with that, move your project forward, start a discussion with them, I'd like to do that. I always call this my shield. So these are the companies that support us all year round. And so our members, this is really what we're doing. The companies that are E2 Tech members are very involved in the PFAS forever chemicals world, in remediation and testing, monitoring, you name it, opportunities for solar siting on brownfields, grid modernization. What's going to happen when we're plugging in electric cars, when we start plugging in electric trucks? What then? Aquaculture and offshore wind are sectors that our member firms are involved in as well. And so if you're working in those areas, I'd love to have you join, or maybe I can make connections with other members. That's really the way that we all benefit. Here's some new members. Clark Energy makes a hydrogen powered engine, which is pretty cool. Key Capture Energy is a New York company that does battery storage. And I mentioned the startup world. I want to spend some time on that. We've got a nice affiliation with Maine Angels. We are known for our panels and our forums. We do tons of those, as does BioME. We try to knock one out about every 3 or 4 weeks. We make them a lot of fun. We're celebrating our 20th anniversary in May. And these are some ones we've done in the past, just to give you an idea. We try to have fun or whimsical titles. We don't always get there. But I wouldn't be doing this right if I didn't flog a couple that we have coming up. So achieving Maine's climate policy goals. We've got some great speakers from this one, including from NYSERDA, which is New York's basically efficiency Maine, and also from the Northeast states, NESCO, which is a utility committee. So you hear about all this climate change and electric grid stuff. What are other states doing across New England? That's a perspective that we're hoping to bring here. And we've got a great under 40 group getting together as well coming right up. We're calling this YEEP, Young Environmental and Energy Professionals. And that seems to be coming together well. So quickly moving towards a closing here, but we have recently surveyed members of our board and program committee to ask them what they think is important coming up. And the response was really heavy on emerging clean energy technologies. So these are some new members that we have. Tanbark is hosted at the Rue Institute and is making an alternative to plastic packaging. Running Tide, you may know, has got a carbon sequestration technology involving kelp. Maine Standard Biofuels is not a startup at all. They've been around almost as long as we have, but this company is really growing. I use their heating oil, which is based on used cooking oil and other bio sources. And these last two are rocket companies. Suspect you've heard of Blue Shift Aerospace using biofuel for rockets, but also Prime Lightworks is a little bit lower profile, but also doing big stuff in space. And this is an electric powered rocket. So those are the areas that we seem to be getting into. I think one of the reasons for that is we have, and I'd love to emphasize this, a strong affiliation with Clean Tech Open, which is a national accelerator. And our regional hub is based in Boston. Really, the takeaway for you here is we have a lot of connections to the kind of 128 loop money, so to speak. So if we can help you bring your idea in any sector, biosciences, you name it, into that world of Clean Tech Open, we'd like to do that. We have funding to do that. And CTO, Clean Tech Open is a pretty broad opportunity. So this is my final slide. It's always my job to flog our upcoming events. Thank you for listening. And I hope that you can make one of these events. And if you see that purple box on the lower right, we have opportunities for you to present just like this. And all of our ideas come from member companies. So if you've got something that you'd like to tell the E2 Tech community about, our program committee is all ears. And thank you so much for the opportunity to present about E2 Tech.
Thank you so much, Marty. I've been to some of your events and I can attest that they're really good events. And even if you're not in that world, they're really interesting and apply to our daily lives. And also, one of the reasons why I wanted to have Marty here is because there is definitely a trend where bioscience spills into other industries. And we're seeing that with aquaculture, we're seeing that with food, but we're also seeing that with clean tech. And actually, when we do our showcase, which is a competition for students, we see that a lot of students do biotech research that has to do with cleaning either water or, removing microplastics and things like that. So there's definitely a lot of overlap, and I think we'll see more and more of that. So thanks again. And now we're going to move to our next speaker, and that's Michael Wing, the co-founder and president of Accelera. I'm very excited to have you, Michael.
Well, good morning, and thank you for the introduction. Thanks for the invite. I'm new to the bioscience group, so it's a pleasure to virtually meet everybody. So I guess I'll share my slide deck here.
Perfect.
And here we go. Okay. So Agnieszka mentioned that I'm a co-founder of a company called Accelera. And Accelera is a company that has developed a wearable technology for improving neurological function. And the science is based on research that came out of Harvard University, the Wyss Institute, BU, North Carolina research. And we licensed this out of Harvard, and it uses a phenomenon called stochastic resonance, which acts like both an amplifier and a filter to neurological signals. And we took this science and over the last several years, we've developed the wearable, and we've been able to get through some clinical testing with some really remarkable results. So if you can imagine, we have this technology that can improve the life of both children and adults with neurological deficits. And some of these deficits, we put them into three categories. We've got the chronic neurological challenges like cerebral palsy, neuropathy, spasticity, balance and gait, things of that nature. Then we have a population that is dealing with rehab, where it's not chronic, but they're on the path of getting better. Things around, knee and ankle injuries, back injuries, things like that. And then, of course, when the last population would be performance. So active adults, athletes, people trying to improve their game, so to speak. And we've got some studies running in all of these. So just to get things started, what I have here is a video of a 3-year-old boy down at the Children's Hospital, Richmond, Virginia. This is one of the subjects in our study. And this little guy, Aiden, has cerebral palsy. It's affecting his lower extremities. And this is a first attempt using Accelera's technology. And you'll see on the left, he has nothing on his ankles. On the right, you can see these bands, these prototype bands on his ankle. What you're going to see here is Aiden's very first steps, independent steps. This is the first time he's walked unassisted in 3 years. So it was this was the results that we're getting in virtually all the children that were tested and some of the adults. So as I mentioned, this is based on Harvard's research and development over time. There's some just wonderful people with great credentials that have discovered this and did some research on this. It's been published, widely published. It's peer reviewed. There's an incredibly large market that we can address. It's a proven platform. We have the hardware, the software, the intellectual know-how, and we have exclusive licensing for the entire body for braces, devices, garments, etc. So what this does is it improves the control of the body, enabling better motion. It allows the body to act more naturally. This is not an artificial signal that's being delivered to the body like a TENS or an E-stem type of application. This is a very gentle vibration working to this stochastic pattern. And we adjust it to a threshold of the individual, and then we can approach these three different markets that I was mentioning. We have the chronic neurological issues. We've got the recovering from joint injuries and back injuries, and then people seeking to improve performance. Just to give you a simple idea on the performance side, they did a study. They took a couple of tests out of the NFL Combine down at Harvard, took some Harvard athletes, ran them through the tests, and they were to improve the population's performance by a small fraction, but the small fraction represented the difference between the eighth place and second place in the NFL Combine. So you can see that those incremental gains at that level are highly important, and that pales in comparison to, a child being able to walk or feed themselves. It's just really, it galvanized the whole team when we started seeing these results. So we're dealing with a very large market, and the big question was, where do we start? Do we start, because you look at these numbers, the balance and gait market alone is close to 50 million people. And these are just in the United States alone. And then we have the injury market and such. And, the challenge that we're looking at over the last several years is selecting that first market, because we can't go after everything at once. We needed to develop clinical models, clinical studies. The cost of those studies are very high. We've burned up a lot of time writing SBIR grants. I guess I'm not the first one to say that in this group, that there's a tremendous amount of work that goes into those grants, and a big learning curve for the business side, and developing a marketing strategy to go after this, and then recruiting the right sales and marketing team. And we've been able to get through all of this. So we found our beachhead, and the beachhead is cerebral palsy. We have a physician champion down at the University of Virginia, Commonwealth University, at the Children's Hospital of Richmond, Virginia. He read through all of the white papers. We sent him some prototypes. He did some preliminary testing, got some really interesting results. And then we set up a study. We found some funding, and I want to credit the Maine Technology Institute. We wouldn't be here today if not for MTI, because they've been incredibly supportive. And so, now we have ties to a major research hospital. We have parents and children and patients that are interested in the technology. They're asking us, when's it going to be available? We've had great positive outcomes. We're setting up future clinical studies. We're starting one in November, and we'll probably later in November be a third one down in Florida. And that technology has gone into practice at these clinics. They have used these devices through the study. We've left the devices there with those clinics, and they're using it on a lot of different issues and seeing good results all across the board. So this has allowed us to develop our technology, develop our device, our first generation. We have our manufacturing partner, which is a company called Saunders Electronics, who's located here in South Portland. I'm a real big proponent of building a good Maine company, working with good, great Maine companies. So Saunders is one of those. They're partnered with us now, and we've gotten through our FDA registration. We have a full quality management program in place. We've been able to recruit the team. We competed in an international business plan for children's health. We won that. And now it sets the stage to go after the larger markets, and we're now positioned for launch. We're actually taking sales orders right now, and we're in the process of building our first inventory. So what's interesting about this is it doesn't take time for this to take effect. You put it on and it works. It promotes the natural body movement. It's comfortable, it's safe, you can wear it continuously. You can wear it continuous versus the episodic sessions that you would see in a clinic where you use it once or twice a week. These people can take them home, have their own personal device, wear it all day long in their therapy, their exercise, their daily routines. Users are reporting more self-confidence. The parents and patients are lining up. The videos demonstrate clear patient, change, sparking other clinical interests. The doctors, the physical therapists, occupational therapists who have been working with this, they've been working across different, different issues like essential tremors and spasticity and balance. It's really fascinating to see how this is all starting to, develop. So in short, it does three things. The study showed that it does three things. It improves the ease of movement, excuse me. It improves accuracy and it improves the speed of movement. So here's a snapshot of how we're selling this device. We're going to sell it in two forms. We're going to have a clinical kit, which we will sell to physical medicine rehab hospitals and physical therapy and occupational therapy clinics. It has multiple devices so that, both, large and small sizes, they can use it on their patients. And when they see results, they will refer the patient directly to us, and then we'll sell them the personal device, which you see on the right-hand side. This is a business model that the team has used. I've got two great people that have joined the team in the sales side. They have experience, combined probably 70 years of experience between them. They have used this model. They have been selling and building and selling devices in the neurological space. They've managed national sales teams, so their confidence is very good. And coincidentally, that improved mine. So this device operates with an iOS app through a smartphone, and we can adjust each device specifically, and the app will run 4 devices at once. So if someone were to need, let's say, both wrists, both ankles, for example, we can do that. We also have the capability of designing and building custom applications, which we've already done several prototypes. One example was an 18-year-old girl who has severe spasticity in her lower back. And as a result, she had an extreme curvature of the spine, and she's wheelchair bound. They were looking at the next option was surgery to install infusion pumps for delivering drugs to the back. So they decided to give this a try as a last-ditch effort, and it worked. It started bringing the spasticity down. So we built a custom wiring harness that we put into a garment they provided, and she's using that now. They just put her through an MRI several weeks back and discovered that the curvature of her back had been reduced by 30%, which, that's pretty fantastic from our point of view. So, the short of it is our technology does work. We have the team and expertise to execute, and our product is ready for market, and we've begun selling. We've been, getting demos and visits with major hospitals. They're setting up their purchase orders, so it's a pretty exciting time for us. We're finally crawling out of the valley of death. so everything's looking very positive, and that's Accelera. So this wasn't as technical as the last, but more on a business perspective. But we're very excited about getting out there, and some of the companies that I want to thank is, of course, the Maine Technology Institute. We get the Maine Venture Fund that's working with us, Saunders Electronics, Flowfold, a small textile company here in Grohman, Maine. So we've got some great partners, and we're looking forward to seeing some real growth. So that's it, and I'll try to field any questions people have later.
Thank you so much, Michael. This is so heartwarming, and congratulations for bringing your product to the market. And I think you have a great story working with Saunders Electronics and Flowfold all around Maine business. So thanks again. And now we're moving to our last speaker today. And our last speaker is our very own Billie Cary, who's our education programs manager. We were fortunate to hire Billie back in the spring to take over most of our educational programs, which have become pretty big. And Maine Bioscience Day is one of the biggest, and it's coming up. So I thought it would be a great opportunity to have Billie introduced to our community. And Billie, whenever you're ready.
Okay, hi, my name is Billie Cary, and as Agnieszka just said, I'm the newest member of the BioME team. As the education programs manager, I get to head up our scholarship and grant award processes, our student showcase, which is open to high school and college students, our middle school summer camp scholarship, and Maine Bioscience Day. All right. So Maine Bioscience Day is a statewide event that aims to get middle school students excited about science. This event focuses on educating Maine's youth about the different career paths and opportunities that are available in the life sciences industry in Maine. Professionals in these fields volunteer to visit local middle school classrooms to talk with students about their career in science, their daily tasks, and the opportunities that are available in such careers. Bioscience Association of Maine started this initiative in 2016 to promote science as a subject in schools and create student awareness for a potential career path in a science field. So this event has increased in participation from 14 schools all the way up to 52 schools last year. We reached over 7,200 students. The last 2 years, due to the pandemic, have been remote programs featuring videos of Maine life science professionals and a hands-on activity. This year, we're going hybrid, with most schools still doing virtual and 15 schools participating in person. So these are the companies that are currently volunteering. Check out those logos. Is your organization not on the list? It might be a great opportunity for you to participate. So here are some examples of what volunteers presented in the past. Patrick Breeding of Lobster Unlimited LLC was a volunteer in Bangor. Patrick presented on varying topics about biomedical engineering in a way middle school students would find interesting. The topics ranged from using photocatalytic nanoparticles for degrading environmental pollutants to the use of cellulose nanofibers derived from the pulp and paper industry for the creation of artificial bone implants to fluorescence microscopy, which is a word that always trips me up, for analyzing cellular state in anti-cancer studies, to using proteins derived from lobsters for skincare applications. So he talked about a whole bunch of different things. Patrick brought samples of nanofiber materials for students to handle and did a quick demonstration on how fluorescence microscopy works by using a black light and fluorescent dyes as an example. Patrick finished the presentations by inspiring students to follow their interests within the world of STEM and how that translates into the real world. So Patrick did that himself. He was a student at UMaine and now runs this company, creating skincare applications derived from lobster. Dr. Catherine Miller Hunt, an assistant professor at University of Southern Maine, was one of 5 volunteers who visited Biddeford Middle School for Maine Bioscience Day. She started her presentation by briefing Biddeford 8th graders about ticks and mosquitoes in Southern Maine, the diseases that they carry, and how to protect themselves and their pets. Students made the connection that these vector-borne diseases had impacted people in their lives. For the hands-on activity, students played a matching game with pictures of mosquitoes and ticks that would be commonly found in the area. The students divided into teams and matched facts that they had learned in the presentation with pictures of the ticks and mosquitoes. So one more example was Jacob Donahue, a student at the University of Southern Maine, was one of the volunteers to visit Brunswick Junior High. Jacob spent time telling students about his studies, his job as a pharmacy technician, and his future goal of attending osteopathic medical school. Jacob also educated students about many non-stereotypical medical professions, such as lab assistants, surgical technicians, phlebotomists, healthcare managers, etc. Since he was in Brunswick, he also informed students that Region 10 High School provides courses in EMT and CNA certification for free. For the hands-on activity, Jacob ran a heart health clinic that discussed the various aspects of cardiovascular health, such as blood pressure, heart rate, diet, and exercise. Near the end of the presentation, students were invited to make a paper tube stethoscope, representative of stethoscopes before the development of the Littmann stethoscope. So these are all examples of what people have presented in the past. So that was our in-person segment. We're also doing virtual presentations again this year, which includes videos from Dr. Colleen Mayberry of Jackson Labs, Dr. Melissa McGinnis of the University of Maine, and Brian Harris, CEO of MedRhythms. These videos delve into the scientists' backgrounds, what inspired them to go into science, what they do in their daily jobs, and how they can do it all in Maine. So at this point, I want to thank our presenters and their companies for giving us their time to create these videos, which will be available to view by the public after the week of November 14th. Along with these presentations will be a hands-on activity distributed to the teachers participating remotely. This year's activity revolves around folding protein channels, including a brief lesson on what proteins are and how they function and why is folding important. And then using origami paper to fold shapes that represent amino acids and combine them as a group into a protein channel. We're excited about this activity that is inspired by Arizona State University's Ask a Biologist. All right, so are you interested in volunteering the week of November 14th to November 18th? These are the schools that are still looking for additional volunteers. Please reach out to me. You can contact me at education@mainebioscience.org. I will put that into the chat in just a minute. Or you can see down along the bottom of this slide, would you like to support as a sponsor? If you'd like to support as a sponsor, please let me know that too. It's $400 to sponsor this program that supports Maine's future life science workforce. So anybody that wants to come talk to middle schoolers, look at this list of schools, please reach out. Let me know if you would like to go talk to a school, and I can put you in touch with the teacher. So thanks so much.
Thank you, Billie. Maine Bioscience Day is a ton of work, so we're very fortunate to have Billie, but also just throughout the years, it's great to see that this program is growing. And I just want to also stress that the overarching goal for Maine Bioscience Day is to build workforce in the future for Maine, because we're going to middle schools to show the students what kind of companies and future workplaces are available, and hopefully inspire a number of students to pursue a career in science. Okay, so now it's time for some Q&A, and I see that our audience is being shy with asking questions today. Sometimes that happens, which is fine. I did get a couple private messages with questions, so I guess we'll start with the one that's out here, and it's to Michael, and it's pretty technical. In light of the changing guidance from FDA regarding QMS, did you focus on 820 or... I don't even know how to say this number for the US lunch, and maybe you can explain that a little bit to our audience as well.
Well, we're actually, we registered with the FDA as an exempt device, right? So we're not making any specific claims. We're just out here supporting neurological function. And to the question, you know, we own the design side, and Saunders Electronics will handle the manufacturing. The quality management system rests on that side of the equation, and we're using the ISO 13485 designation.
Okay, thanks, Michael. I have a question for Dorothy regarding blueberries, and that question is, you mentioned feeding blueberries to the obese rats or mice. How much blueberry was given? What was the percentage of their diet?
Okay, unmute. Yes, it's equivalent when calculated with a human to the human. It's about a cup, but we discovered that even half a cup will have similar effects. So the best way really would be to, have it every day, because the effects, as we saw, kind of last for 24 hours. So it's good to have daily, and just make a smoothie with it very easily. Or have them, the fresh frozen organic is fine, to have it on your oatmeal or, as part of it. So half a cup, between half and 1 cup.
Okay.
Fresh or fresh frozen.
And is it safe to feed your dogs or cats blueberries?
Well, I'm not a veterinarian. I haven't worked with cats and dogs, but yes, I would say, yeah, I would. It's a very healthy berry with all the goodies, the antioxidants, the polyphenols, and beyond antioxidant, anti-inflammatory. So yes, I would say, but please ask a veterinarian for the cat and dog.
Okay, we definitely will. Michael?
A quick question for Dorothy. I think that's fascinating what you discovered of the blueberries. Do you know offhand if there's much of a difference between wild blueberries and the cultivated blueberries?
Yeah, there are differences, nutritional differences, definitely, because the wild blueberry has about 3 times the composition, the content of the antioxidants than the high bush blueberry. Yeah, and I had the table. Yeah, specifics. I can send you some specifics.
Thank you.
Definitely. It's packed.
Right. And someone else also is asking about the difference between wild blueberries and cultivated blueberries. And is it the same, or are there differences?
Yeah, the difference is in nutritional content as far as vitamin C is concerned, manganese is concerned, etc. Many. And I can send that. I don't have my slide with me, but yes.
So basically, wild blueberries have a lot more.
Yeah, yes, antioxidant. And also, so you can, if you are to, I mean, we have seen there are many researchers that work with hibiscus blueberries and have seen repeated these, because I was one of the first ones that started this field on vascular function, and then many repeated that with animals and humans, discovered the same similar effects. So, which I'm happy. And they use the hibiscus blueberries. So also, but
Okay, great.
compositionally, have 3 times the antioxidants that the high bush do.
Okay, thank you so much. I think I'm looking through my questions here.
I can ask Michael a quick one. Michael, I'm in Biddeford, and the stochastic technology, you probably know the team from Propella, which makes, I think,
Absolutely. Yeah. I'm very, very familiar with Propella. Their technology also came out of Harvard. In fact, John and I met down at Harvard when we were doing all this due diligence and on the technology. And what Harvard did is they split the licensing so that Propella is focused on that infant mattress. And when I saw the numbers for what that was achieving for these infants, it was really significant. I mean, it really does work. And but we've got everything from head to toe. So we both work with Saunders, and we stay in contact, and I'm sure John is going to do really great things with that technology.
Yeah, I'll put their website in the chat. That's cool that you guys know and each other and are supporting each other. Yeah.
And you can see if you go to accelera.us, not .com, but .us. We're just launched our new website, and you can start, you can see some of the research and publications that were associated with it, and some of the videos as well.
Cool,
great. Yeah, I'll definitely check that out too. We have a question from Dr. Doug Curry to Dorothy, and it has a lot of words I can't pronounce. So I asked Doug to ask that question in person. So Doug, whenever you're
Thanks, Ignatia. Yes. Dorothy, that was really exciting to see how wide-ranging the effects of these blueberry extracts are. And I was just wondering, do you know what mechanisms are affected by the anthocyanins to inhibit cell migration and blood vessel formation?
Oh,
So
yes,
is it also via the cytoskeleton and the RAC-RO pathway?
Yes, so what we explored, yes, it was the RAC-RO, and we found with the anthocyanins that they would inhibit that, while the phenolics will promote that. Correct. That's what we explored.
So in terms of developing, you've isolated for the skin, you've isolated the phenolics as the key
Yes, the
compound.
yeah.
Are there things that you're looking at for the anthocyanins as possible
No,
key effectors?
no, because the anthocyanins, it has been known maybe 15 years ago, there were a couple of papers that suggested, and they had documented that anthocyanins actually inhibited cell migration. We knew that, and so we didn't go with that. So, but that would be conditions that will benefit with that. For example, cancer, you know, where you want to stop angiogenesis, but we just decided to go with the phenolic to promote, just because that was known, and also for the patent, you know, they would have never been able to give us the patent. So we just went with that. But, you know, when you do the combination, it seems that it promotes, but we just didn't want to touch the anthocyanins.
Yeah, that's very exciting. Yeah, thank you.
Thank you. Yes, I'm hoping. Well, we're just at this point, like Michael, congratulations and all that, because taking all those courses and just trying to decide whether to start a startup or license the technology. So it's like crossroads right now.
Yeah, it's certainly not easy. And let us know if we can help in any way.
Thank you.
As you know, we have a lot of connections with MTI and other organizations in the state. Okay, well, we're nearing the end here. I just also wanted to reiterate that Maine Bioscience Day is coming up next month. We're still looking for some volunteers. I saw Marty, you said that maybe you would be willing to volunteer, so we'll talk to you later about this in Biddeford. And other than that,
I have a question.
Yes.
Can I ask you? I put it on the chat. Michael. Michael, so can you share the cost of that apparatus with us? And the other question was, will insurance pay for it?
Good question, Dorothy. The personal device, which is this right here, right?
Yes.
This device, which is a single device, is retailing to customers for $500. So it's very reasonable.
Yeah.
And then, of course, we've got the clinical kits that we sell at a different number to the hospitals. But we're not pursuing a reimbursement strategy right now. It may be in a couple of years we'll do that, you know, but we really were focused on, let's get this thing to the market, start building brand, get people interested in it, and it'll make our argument a lot better, because we'll be accumulating data as we go. So we wanted to price this in such a way where it was affordable for people, still provides the margins that we need in the business to grow it. And like I said earlier, there's just, once this grabs hold, it's the populations are so large that we don't need a lot of that market to be very successful.
Oh, it's wonderful. Congratulations. It's great.
Thank you.
Okay, thank you so much. And apologies that I missed that question, but I'm glad Michael got the chance to answer it. Okay, so it is 11. I just want to say our next bigger in-person meeting will be our holiday mixer. We're so happy to have that back in person, and that's going to happen in December. So please stay tuned. We may have another coffee hour by the end of the year. If you're not subscribed to our emails, please do so, and then you can keep up with all of our activities and events. Once again, thank you so much for joining us today, and enjoy the rainy day. Maybe it'll get better soon. Thanks so much to all of our speakers and all of our audience today. Bye.
Thank you.